All right, welcome back, fellow scientists. So now that we know how DNA replication and transcription translation are supposed to work, now we want to look at what happens when that goes wrong, right? You can kind of sense a common theme, like all throughout with cell division and with cells and with photosynthesis, cellular respiration, we talked about how it's supposed to work, and then what happens when it goes wrong. Okay, so same kind of thing. What happens when the process of DNA replication or transcription or translation, when that messes up? Okay, specifically, we're going to look at what happens when there's a mutation to DNA. What happens when there's a change to DNA? So you got to have this big picture in your mind. Okay, you got to remember the central dogma of biology, that DNA is used to make RNA, and RNA is used to make proteins, and proteins are used to make you. Right? So it doesn't take a rocket scientist to figure out that if there's a change to DNA, there's going to be a change to the RNA, there's going to be a change to the protein, and then quite possibly there could be a change to you. Okay, so now we got the DNA to RNA, we got the RNA to proteins. Mr. Leo, this is kind of fuzzy here. How in the world do proteins determine me and who I am? Well, we already talked about that proteins are the workers, but I want to elaborate on that a little bit. So go ahead and get a new notebook page, right? Pay no attention to the diagrams right now. We'll get there. But grab a new notebook page, and this is a very, very important phrase right here. This is something that we hammer even in AP biology, right? For proteins, for all molecules actually, shape determines function. Shape determines function. I'm pretty sure that I said that before in chapter 2, but just in case I didn't. Shape determines function. Shape determines function. So in chapter 2, when we were talking about proteins, right, we drew Pac-Man as the protein, and you remember we had the chicken liver lab where we had the paper, the coffee filter disc, and we dipped it in the chicken liver frappe, and then the H2O2, the hydroperoxide, went in, and then the enzyme, the peroxidase, broke it down into water and oxygen is gas, and that made the disc rise, so we could look at the function of the enzyme based on how fast the disc rises. So all that to say, right, shape determines function. So this enzyme, which we're calling peroxidase, has a different function than this enzyme, right? And go ahead and draw this, right? This is key. Shape determines function. So this enzyme has a different job than this enzyme, and this enzyme has a different job than this enzyme, right? So all of these enzymes have different shapes, therefore they have different functions, okay? That's how proteins make you. That's how the workers decide what job they get to do, right? If you have a worker with a hammer, it's going to do a different job than if you have a worker with, oh, I don't know, a skill saw or something like that, right? Different shape, different function, different jobs. So that's how proteins make you. Now, Mr. Leo, how in the world do we determine the shape? Well, that requires a little bit more in-depth explanation. So if we look over here at these charts, which, by the way, you can find on OneNote under your class notes tab. I didn't load all the diagrams when I wanted to with the same page. So you'll see two video 11 pages under your class notes tab. So I labeled this one use this one because that's the one that you need to use. That's the one that has all the diagrams. So you don't have to, but if you want to, if you're OCD and you want to make sure that your notebook looks exactly like my notebook, then you can do that. But basically, this chart simply lists all 20 of the amino acids. Well, Mr. Leo, I thought we were talking about proteins. Why are we talking about amino acids? Well, remember that amino acids are the building blocks that make proteins. And remember that amino acids, they all have the same backbone, right? But then they all have different R groups. And these R groups have different properties. So we have hydrophilic R groups. Some are positive, some are negative. We have uncharged R groups. We have some special cases, which we're not going to get into. And then we have nonpolar or hydrophobic R groups. So all of those have different properties. Okay, so this is what DNA, RNA does to make proteins. They tell the ribosome which amino acid to put in which spot that makes the chain that makes up the protein. Okay, so here it is kind of visually, right? Come on, move down. There we go. Okay, so each of these little rectangles right here, that represents one amino acid. Okay, so remember talking about the primary structure for protein. So the primary structure is the sequence of amino acids, which is determined by the RNA, which is determined by the DNA, okay? But then the primary structure that determines the secondary structure, the order of the amino acids, which amino acid goes in which position, determines whether the ribbon folds into a alpha helix or a beta pleated sheet. Okay, and then the alpha helix, beta pleated sheet, and the location of those, and the primary structure determines the tertiary structure, which is the three-dimensional shape of one chain. Some small proteins stop here. They don't have a quaternary structure. They just have one chain. Some proteins like hemoglobin, that's the classic example, that's the one that's shown here, have 4 separate subunits, 4 separate chains, and so they actually have a quaternary structure. But suffice it to say, all enzymes, all proteins have a three-dimensional shape, right? And that three-dimensional shape is determined by the sequence of the amino acids. And that sequence is determined by the RNA, and that RNA is determined by the DNA, right? And so all of that leads into the shape, and then the shape determines the function, right? So it all like, that's the key. Shape determines function, okay? So now, let's go ahead and let's look at an example. Dun, 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 dun. Perfect. Right here. So you may remember this or something similar to this from Mr. Anderson when we talked about the decoding rectangle. Okay, so we're going to get some practice using this in class, but I thought that I'd give you a little practice here in the video too. So here is 1/2 of a DNA double helix, right? If you're OCD and you want to write in the other half, that's great. Oh, and by the way, you can also find this graphic on OneNote under your class notes tab in the video 11 diagrams. Use this one. Page. Sorry that it's complicated. It's biology, whatever. Get over it. Okay, sorry, that's kind of mean. I didn't mean to say that. So this is 1/2 of a double helix, right? So if you want to write in the other half, that's great. But what I would like everyone to do, regardless of if you're OCD or not, is to make the mRNA that would be transcribed from this DNA. So go ahead, pause the video and do that. Okay, perfect. So hopefully you came up with something that looks like this. Now, in videos past, we were talking about transcription and translation. We recognize that mRNA is broken up into groups of 3 letters called, yep, codons. Perfect. So we're gonna go ahead and do that. We're gonna break these up into groups of 3 letters called codons, just like that. And so now that we have that visually broken up, now we can use our decoding rectangle, okay? And we can this is how the decoding rectangle works. We take our first nucleotide, and it's very clearly labeled right here, first letter. So first letter is A. So that means that the amino acid, right? These are the names of the amino acids: threonine, asparagine, leucine, aspartic acid, glutamic acid, blah, 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 blah. Okay? So the first amino acid could be anywhere in here. Okay, and then the second letter is labeled up here. So second letter is U. So AU, so where those intersect, now we could either have isoleucine or methionine as our amino acid that starts the protein. So then we need our third nucleotide. We need G. So G is right over here, and so AUG, where all of those kind of come together and intersect, it says methionine. So what I've done is I've just kind of abbreviated it, methionine. Okay, so we'll do one more together, and then you guys are going to do these last 2 on your own. So CGU. So CGU, so C is the first letter, anything in here. G is the second letter, so here's the intersection. Now this is kind of handy because there's just one amino acid that it could be, but if we really want to make sure, then we can say U, yep, CGU, perfect. Arginine, excellent. So then that's arginine. So go ahead, pause the video, and then you guys do the other ones. All right, so hopefully you got this. Met, arg, thir, stop. Now, stop isn't actually an amino acid. There's not an amino acid named stop. So that codes for a release factor that attaches to the mRNA, and then like it explodes the ribosome. Like it makes the ribosome, you know, go away and stop translation, and stop translation. So this isn't actually amino acid. It's just instructions to the ribosome to actually stop. Okay, so that's background. Now let's actually get into mutations. All right, so mutation, go ahead and write down. Mutation is any change to DNA. I'm going to go kind of quickly because I'm running out of time. Point mutations are changes to one or very few nucleotides. So make sure that you have that written down. I would like to distinguish point mutations versus chromosomal mutations. Chromosomal mutations we already talked about. That's where like whole chromosomes we have extra or we have too few. So we already talked about Turner's and Klinefelter's and trisomy X and Down syndrome, which is trisomy 21. So we're not talking about that. We're not talking about huge changes to the chromosomes. We're talking about changes to one or very few nucleotides. Okay, so in class, we're going to actually use a decoding wheel or decoding rectangle rather and DNA, and we're going to transcribe it to mRNA, and then we're going to translate it. We're going to use shapes to represent amino acids. And it's going to be great, right? But this, we're going to start out a little bit simpler. We're going to use a different kind of analogy. Okay, so I'm kind of saying that this is the mRNA. Okay, and we can break the mRNA up into codons, just like this. And I need to move over here. Perfect. Like that. Excellent. And so then these codons, they can kind of be thought of as like the amino acids, and then the sentence can kind of be thought of as like the protein. It's kind of like that Flynn scientific decoding sentences activity that we did in class last week. But anyways, so the cat and the hat, which I know I have kids. I read this almost every day. That's not the actual name of the book, but that's okay. I just needed a sentence that made sense with, you know, all three-letter words. So just work with it, okay? So go ahead and write that down. That's normal. That's the normal DNA before any mutations have happened. So now we're going to change this in a couple different ways, and we're going to look at what would happen. So go ahead and write down substitution. We have two types of substitutions, neutral and non-neutral. So we're going to go ahead and use our analogy to look at this. So first of all, we are going to have a non-neutral substitution. So here's the new mutated mRNA, which was made from the DNA, right? And so we're gonna break this up. Da-da, da-da, da-da. And of course, you can see that we changed the C to a B, and it's no longer the cat in the hat, it's the bat in the hat. So that actually changes the meaning of the title. Okay, it's no longer the cat that's coming in with thing 1 and thing 2 and messing around with the fish and everything like that. Now it's a bat, which is still a mammal, but it's a different mammal. It has wings and it doesn't have a tail, I'm pretty sure. So anyways, that is non-neutral. That changes the, if we extend this analogy, that changes the enzyme, that changes the protein, that changes the structure, that changes the function. Okay, so we could also have a neutral substitution, right? So we could do this. We could do something like this, like that, and we've replaced our C with a K, right? And so the cat and the hat, if you spell it phonetically, it still works. It still makes sense. So that's a neutral substitution. It doesn't change the shape of the protein. It doesn't change the function of the protein. Okay, so that's a substitution. Now the other types of mutations that we have are called frameshift mutations. Okay, and there's two types of frameshift mutations. There's insertion and there's deletion. So let's go ahead and look at insertion mutation first. So here's our original sentence, right? And here we've changed something. Can you guys spot the change? Yep, that's right. We added an A. Okay, so normally there's not an A between the N and the D, but here there is. So let's go ahead and break this up into our codons. Huh, that's weird, right? So the cat is, but then we inserted, and so that changes all the codons that are downstream of the insertion. Okay, so that's why it's called the frame shift. So breaking it up into groups of 3 letters, that's called the reading frame. And if we insert or if we delete, we're changing the reading frame. We're shifting the reading frame. So that's why it's called a frame shift mutation. So that's what happens with an insertion. And then a deletion, we can go ahead and look at that. So see if you can spot the deletion. Yep, that's right. We deleted the C. All right, so here again is our regular sentence, which we can break up just like this. And then if we look at the deletion, we can break it up just like this. It's like, huh, okay. So that's upstream of the mutation. That's still the same. But then if we deleted something, then the ribosome still reads the mRNA in groups of 3 letters. And so that shifts the reading frame, and the rest of the sentence doesn't make sense. Similarly, the rest of the protein is not going to make sense, and so it's going to be a non-functional protein. So this has been Mr. Leo. Today we have talked about mutations and what happens when something goes wrong with DNA. Thank you so much for watching, and I will see you on the flip side.